This video is going to be on the 3 general causes of anemia. But before going into the causes, I have pictured here normal red cell physiology. Red cells are produced in the bone marrow, circulate in the blood vessels to deliver oxygen to the tissues, and then at the end of their lifespan, the red cells are taken up by splenic macrophages. Most of the components are recycled, but the waste product bilirubin is excreted back into the blood, and it makes its way to the liver, where it's excreted into the intestine and into the feces. Okay, so now for the 3 general causes of anemia. The first is hypoplasia or aplasia. That's a problem with the bone marrow production of red cells. The second is hemorrhage. Whole blood is being lost from the blood vessels. And the third is hemolysis. There's increased red cell destruction or lysis. So going back to hypoplasia and aplasia, there is decreased production of red cells by the bone marrow, and this results in a non-regenerative anemia, meaning that the reticulocyte concentration is normal or decreased. Now, depending on the cause of the anemia, there may or may not be additional cytopenias. So there may be decreased concentrations of neutrophils, neutropenia, and there may be decreased concentrations of platelets, thrombocytopenia. For hemorrhage, that is when we're losing whole blood from the vessels into the extravascular space. And to support hemorrhage on our CBC, we also look for hypoproteinemia because there's lots of protein in whole blood, and that is also lost when there's hemorrhage. So decreased concentration of protein will support hemorrhage. Now, hemorrhage can be pre-regenerative if it's within 2 to 3 days of the onset of the hemorrhage. But after 2 to 3 days, you would expect the regenerative response to be seen in circulation, so you will have a reticulocytosis past 2 to 3 days. And then, if there's chronic external hemorrhage, so chronic external loss of iron from the body, then that can result in a non-regenerative anemia because of iron deficiency. Now, to localize the source of hemorrhage, we can look to our serum chemistry or plasma chemistry. To identify GI hemorrhage, we look for an increased BUN relative to creatinine or increased blood urea nitrogen relative to creatinine. And that's because when there's GI hemorrhage, the protein that's lost in the GI tract is resorbed. So it's essentially like a high protein diet. More protein absorption, more protein conversion to urea, higher plasma BUN concentration. To identify urinary tract hemorrhage, we look for red or dark urine grossly, and we also can look in the microscope at the urine and see intact red cells in the urine. This is known as hematuria. Now going to hemolysis, that is when there is destruction of red cells either within the blood vessels or outside of the blood vessels within macrophages. If they occur within the blood vessel, so red cells are being lysed within the blood vessel, then this is called intravascular hemolysis. And the red cells are lysing, they're releasing their hemoglobin content into the plasma, and that gives the plasma in the serum a red discoloration or pink discoloration. This is called hemolysis grossly. Now this hemoglobin, if it's at high enough concentration, it just doesn't stay in the plasma. It's actually excreted into the urine or overflows into the urine. So you can have red or dark urine with hemoglobinuria, hemoglobin in the urine. The other form of hemolysis is extravascular hemolysis, and this is when the red cells are being destroyed by macrophages. So there's increased phagocytosis of red cells by macrophages. Now, this results in increased degradation of red cells, more bilirubin production, and if bilirubin's at high enough concentration, then that will turn the plasma in the tissues a yellow color, which is known as icterus. On our serum chemistry or plasma chemistry, we can also look for increased bilirubin concentration, which is known as hyperbilirubinemia. This type of anemia can be pre-regenerative if it's within that 2 to 3 days of onset of hemolysis, but beyond 2 to 3 days you would expect to see a regenerative response, so you will see a reticulocytosis. So that's it. The 3 general causes of anemia are hypoplasia/aplasia, hemorrhage, and hemolysis, and we distinguish these general causes of anemia based on the regenerative status, presence of reticulocytosis or not. We also look to the protein concentration and the color of the plasma in the urine.